thank you to all who are able to very adeptly switch over to the new and improved Zoom platform that can accommodate the great level of interest that we saw for this program tonight. So much appreciated for that. Quick note of introduction, this webinar is part of the Narragansett Base Samp webinar series. So the University of Rhode Island's Coastal Resources Center, Rhode Island Sea Grant, and the Rhode Island Coastal Resources Management Council are proud to offer this series. It is designed to be a complement to the Narragansett Base Samp aquaculture element process that is underway. Many of you have been involved right from the beginning for the last few months in various stakeholder meetings, additional webinars, etc., working group meetings. So we're grateful for your participation in that. And again, this is a series that is designed to really kind of pull some of those topics that have come up in those stakeholder meetings and delve a little bit deeper in an evening hour sort of format. So again, these are all posted. The slides and the recordings will be posted on the Base Samp website that's right here on the screen. And without further ado, I wanted to quickly introduce our topic for the evening, Shellfish Restoration and Enhancement in Rhode Island. We will hear from several wonderful folks in the state. Robbie Hudson is the Shellfish Hatchery Manager. Susanna Osinski is the new Shellfish Technician at Roger Williams University. Robbie is also at Roger Williams. Ed Cabral is the Harbor Master for the town of Warren. And Eric Schneider is a marine biologist at the Division of Marine Fisheries, Rhode Island DEM's Division of Marine Fisheries Department. And we are so grateful to have the expertise and skill and savvy of Skylar Bayer, a professor, assistant professor of biology, aquaculture, and an extension specialist at Roger Williams University. She will be facilitating the discussion portion, and I will turn it over to her to in more detail share the speakers' bios. But before I do, one last note and reminder is just if you could all please put your questions in the chat. And a lot of folks joining us today. We will keep track of those and bring those up after the presentations. And oh, sorry, that says Julia. Sorry, that should be Skylar. I apologize. And then of course questions will be addressed. Any ones that aren't, we can forward on to the right person afterwards and get the responses back to you in a timely manner. So with that, Skylar, I will turn it over to you.
Thanks, Azure. It's nice to be here. For those that don't know me, I'm relatively new in the Rhode Island area. I started my job here at Roger Williams University during the pandemic. It was mostly remote last year, but I'm an assistant professor of biology, aquaculture, and extension. And I'm really excited that part of the presentations today will be from our team, the Shellfish team, Rob and Susanna. So I'm looking forward to that. And as Azure said, please put your questions in the chat box. Try to keep yourselves muted during the presentations. And then at the end of the presentations, I'll facilitate the Q&A. I'm looking forward to some of the questions folks may have. So first presenting will be Eric Schneider, who, as Azure said, is a principal marine biologist with the Rhode Island Department of Environmental Management's Division of Marine Fisheries, and he coordinates the oyster restoration activities for Rhode Island. He's also leading the state's Shellfish Restoration Plan that will begin early next year. He's also a principal investigator for several research programs, including work on habitat protection, acoustic telemetry, and he's also a PhD student at Northeastern University. And he's on several different management committees representing Rhode Island, all sorts of different fish, as well as estuarine and marine habitat management activities. Following that, Ed Cabral is the harbor master for the town of Warren, and we'll hear from him for a couple of minutes at the beginning of the second presentation. And then we'll hear from Rob Hudson, who is our hatchery manager here at Roger Williams University. He's been part of our shellfish research group since 2016. And before that, he spent over a decade working with Save the Bay on bay scallop restoration as an ecologist. And he is also currently a PhD student at the University of Rhode Island with Marta Gomez-Chiari. And then Susanna Ozinski is our newest hire at Roger Williams University on our Shellfish team. She's our field and extension technician and runs the farm. And she recently graduated from Cornell University with a degree in biology and business. And I think most interestingly, she grew up on an oyster farm on Long Island. So she has basically over 20 years of experience growing oysters with her family. We're really excited to have her join our team this fall. So welcome Eric, Ed, Rob, and Susanna to the webinar. And to get started, we're going to start with Eric. So Eric, when you're ready, we can begin with your presentation.
Great. Thank you so much, Skylar. And thanks everyone for making time to join today. Just give me a moment while I bring up my screen. Can you guys see my presentation?
Yes.
Oh, great. It's not showing on my end, but I'm happy you can see it. That's all that really matters. And so, Skylar, if my audio goes in and out, perhaps I could ask you or Azure just to interrupt and let me know. And that way I can try to fix it. But without further ado, thanks again, everyone, for making time thanks, Azure, for joining today. I'm going to jump right in because I have a lot of ground to cover. In Rhode Island, eastern oyster reef habitat is primarily found in subtidal waters in our coastal lagoons and estuaries. And when intact, oyster reefs provide an array of ecosystem services, including nursery habitat for fish, crustaceans, and other invertebrates. They can enhance water quality by removing excess nitrogen. They can be used to stabilize eroding shorelines, and also can provide harvest opportunities for food or other social benefits. It's been well documented that present-day oyster populations have declined to roughly 15% of their former global abundance, largely due to overharvest, disease, habitat degradation. And similarly, natural oyster populations in Rhode Island have been severely depleted, with current estimates at roughly less than 1% of historic levels. Thus, this is one of the reasons why restoration is needed to restore these wild populations, as well as some of the ecosystem services that have been lost. Today, I'm going to focus on two oyster restoration programs. One is aimed at restoring wild populations. The second is aimed at enhancing fish habitat. And I'm sure no one's going anywhere, but just make sure you stick around for the next talk. I'm definitely looking forward to hearing it. So the first program I'm going to talk about is the Rhode Island Nrcs EQIP Oyster Initiative. And EQIP stands for Environmental Quality Incentive Program. This is a partnership. It's administered by Nrcs with assistance from Rhode Island's Division of Marine Fisheries and our Rhode Island aquaculture community. The goal is simply to try to create sustainable wild oyster populations. I'm only going to focus on what I would call the more recent work from 2015 on. Generally speaking, this work has targeted 7 different locations. 40 different aquacultures have contributed to this work, and I'll talk later on how monitoring and incorporating research has been critical in our ability to further this program. And just to give some perspective as to how the Division of Marine Fisheries comes in, we're primarily responsible for determining where restoration will take place, as well as the techniques that are going to be used at that specific location. Overall, we lead the management aspects and permitting aspects. And one of the things that is critical, really a critical first step in determining the overall success of an oyster restoration project is the siting of that project. On the right, you'll see a figure from a TNC report. It's showing an oyster habitat suitability index. And I know the legend is busy. All you really need to capture in this is that the cooler colors, the blues is poor. And as that heat gradient increases, as you get red, that's better. Those are the most suitable habitats. And so this type of model can be really useful. It can incorporate a number of physical and environmental variables to give support as to what habitats are most suitable for oyster restoration. Unfortunately, the current models are spatially and to some degree data limited, but when they're available, the division certainly incorporates them into our planning decisions. We also lean heavily on subaqueous soil maps, any measured or inferred salinity levels, as well as the historical occurrence of oysters and best professional judgment. Sometimes that comes from aquaculturists, shell fishermen, and other staff that spends time on the water. I'm going to walk through for folks who aren't familiar with the actual restoration process. I'm going to just give you a quick overview of what that looks like. One of the practices or restoration scenarios, we could call it, that's used is when the substrate for larvae to settle upon is limited. So there's ample larvae in the water, there's just not enough substrate to support the settlement and recruitment of that in the system. And so in those cases, we can add shell, or also known as culch. It was originally surf clam and it's shifting more to oyster shell work, but that can be used to enhance the habitat so settlement can increase. The more typical practice, however, given the status of oysters in Rhode Island, is to create seeded reefs. And so we use these when their brood stock is insufficient or there's just not enough larvae in the water to settle. And so as you saw before, we would create this culch reef base, but then we add on top of it that these reefs are seeded with juvenile spout and shell oysters. And this picture in the top right shows what a spat and shell oyster. It's where you have several spats settle on a single piece of shell, and that's what you would find in the wild, and different often than what you would see at a restaurant where you're getting individual oysters. And so the picture on the right shows what a much larger version, but this individual oyster, and we're able, through some of the work, I'll discuss it in a few minutes, but now we're able to target different size classes depending on the site and scenario. Often the oysters we put out as spat and shell are very large, but through this work, we've migrated all the way through to using large single oysters. One of the most important aspects, really in terms from the division perspectives, but really the overall success of this partnership, is monitoring. It allows us to get feedback on the practices that we're doing, and certainly improve those restoration. And that's not new to anyone. But I would like to offer that through having monitoring really on all of the oyster restoration work that's been conducted recently, so since 2015, we've been able to dramatically and continually improve these practices. And now we're at a place where now we can modify the technique for a given location. And that might sound very basic or logical, but when this started, we were really using, a 1 or 2 size fits all options. But now we've moved to a scenario, really like a palette of different scenarios where we can really tweak it based on what the data suggests would be best for that system. And this has allowed the Rhode Island Nrcs equip approach to become really the standard for the New England region. And it's also gaining coastwide attention. Folks from Texas to New Hampshire, from end to end, have been meeting with NRCS in Rhode Island to learn more about their program and how they can implement it or implement a locally modified version in their states. However, even with all of these advancements, recruitment is still not sufficient to support self-sustaining oyster reefs in most of our systems. And we'll revisit this later. But I have to quickly move on to the second project that I'm going to talk about today. Remember, this project is a fish habitat enhancement project. It's a different goal. This is a partnership between, again, Division of Marine Fisheries, the Nature Conservancy, and Northeastern University. And simply put, the goal was initially to evaluate whether the creation of oyster reefs can improve juvenile sport fish populations. Work has been done in other regions, in particular the Mid-Atlantic and the Gulf, that has shown this is indeed the case, but it had not been replicated in the Northeast. And so we took that on. In a nutshell, the process was to establish baselines of what the fish community looked like in these systems before making the reefs, go out, create some oyster reefs, and then continue to monitor that community afterwards to assess what the changes look like. So on the map is a right, don't get bogged down with the details. You'll see some areas just popped up that are highlighted in red. And if we just focus on those, I'm going to narrate you through the elements that are important. There's more information than what you need here. But this work was conducted in 2 coastal lagoons, coastal ponds, Quanakatog Pond, which I'll refer to as Quani, and Ninigrit. Right now, I'm just highlighting the Quani pond. And all I really want you to focus on is that this work was conducted in 3 different regions. You can see 1 in this left-hand panel and then 2 in the right. And within each region, we maintained a control plot. So that's a plot where we did not build a reef. It just stayed barren sand just as before. And we also monitored the 3 reefs that we created in this scenario. We had a similar design, but slightly different in Ninigrit. Here we had 2 regions, a northern region and the southern region. And within those regions, we had also, like in Quani, a control plot where there was no reef built, but then we built a reef that we did not seed, and we built some reefs that we did seed. And for all of this work, we monitored those oyster reefs at least once they were built, twice a year in the spring and the fall. And then we continued to monitor those fish communities monthly, annually from April to October. And this work is still ongoing. The data I'm going to show is restricted to the years that you see above. And so to our initial charge of whether or not we can have a response, a positive response of fish to creating the oyster reefs, we found that reef-dwelling species like tautog, black sea bass, cunner, for these species, that fish abundance was increased after reef creation. Sorry, my text got truncated there, but that was compared to before reefs. I don't have a lot of time. I'm just selecting one figure to show you to really kind of illustrate this change. So to orient you, we have catch on the y-axis or the left-hand vertical axis. We have years going across the bottom. In Quani, we monitored that. All of these sites in 2016 before we created fish, and those are actual zeros. We did not catch any fish at these sites. But then you can see once that the reefs were created, we did start to catch fish. And that generally the abundance tracked with oyster biomass or densities. I'm not going to show that latter result. That's coming from really work that's still in progress. As I mentioned, we're continuing this. We're developing a pretty impressive long-term fishery and habitat survey. And now we've focused into developing more density estimates of fish, the macroinvertebrate community, in an attempt to try to quantify the fishery production that these oyster reefs can support. I'll quickly share some other results. You'll have to think back that again, in these ponds, we had these different regions. And when we were going through and monitoring these oyster reefs, we found that mortality did change depending on the pond or the region. This is shown in this figure to the right. On the x-axis here, your horizontal axis, we have natural mortality. And so what obviously you want to see natural mortality or mortality lower. So the dots that are closer to the left, those are better results. So remember in Ninigrit, we had 2 regions. Those are shown up here. We had 3 regions in Quani. Overall, we found that mortality differed not only between ponds, but also within regions. And although I'm not showing the data here now, we also found that mortality can vary between reefs, even within regions. Obviously, there's a number of possible site level factors that can influence mortality, but we also found that the source or the lineage of oysters can also affect the long-term mortality rates of those reefs. So quickly, to give a brief summary of the EQIP and FHE, the fish habitat enhancement work. Overall monitoring has been critical to improving the restoration practices. The Rhode Island EQIP program now is the regional standard for the Nrcs oyster restoration work, and it's gained coast-wide attention. We found that creating oyster reefs in Rhode Island can be used to enhance fish abundance of at least of certain species, and that we're currently working to continue that work to develop production estimates. We also found that oyster survival varies within and between locations. And again, that recruitment is limiting the long-term self-sustainability of our restored oyster reef communities. And so now I'm going to shift into the, where are we going? How are we going to move forward with this? Some of the challenges we need to address include this recruitment and survival issue. And so again, improving recruitment and survival, both on restoration reefs as well as throughout these systems, is really needed if we're going to take that next step forward. And one of the aspects that we need to address is, we need to improve the information available for us to make good decisions about siting restoration work. And so currently we have another project. I'm not going to talk about it other than a couple seconds here, where a partnership with DEM, Northeastern Nature Conservancy, that's supported by the Southern New England program of Restore America's Estuaries, and Rhode Island Nrcs, is aiming to collect more information about recent restoration reefs that have just fallen out of kind of our restoration cycle, as well as wild oyster communities, to develop an updated and expanded habitat suitability index. But in addition, we also need to consider seascape level attributes. These can include metapopulation dynamics, and these can be aspects related to connectivity within or between subpopulations in the system. And this is important depending on project goals to ensure that we can cite a project if we're trying to provide broodstock, or if we're trying to enhance habitat for receiving larvae, that ultimately we need to have a better understanding of these aspects. We also need to ensure that we avoid impacts to other habitats and minimize conflicts with other uses. It's been widely recognized that Rhode Island needs a cohesive, overarching restoration plan. Excuse me. One that considers not only the needs of stakeholders and managers, but also addresses some of the ecological and social data gaps that we have. And we also recognize that we need to improve our outreach and our stakeholder engagement. So in response, DEM Division of Marine Fisheries is leading a partnership that will develop a statewide shellfish restoration and enhancement plan for Rhode Island. We recognize that we're working in a system where relationships between people and the environment are coupled, that these relationships can be complex, and that details are important. And so we're proposing to use a social ecological systems approach to develop our shellfish restoration plan. And these approaches are commonly used to try to better understand these dynamics of natural and environments and how they influence one another. We envision using a process that aims to build public support and awareness for shellfish restoration by engaging stakeholders, assessing their values, preferences, and knowledge. We also want stakeholders to provide information that we can use to help inform restoration priorities and goals. Right now, we're still in the process development phase. You'll see on the bottom, and I'll refer to this in just a minute, you'll see us move forwards. This is not the actual social ecological systems framework. This is just a very simple flow chart of how the process can move forward. So right now we're developing the process. As it was mentioned earlier, come the spring of 2022, we're actually going to have a kickoff meeting. We're going to start to collect information. Over a period of time, we're going to collect information. We'll be able to use certain analysis to develop a draft plan. That plan will be finalized, but even when it's finalized, it will be a living plan. And as new information from research, monitoring, or other sources become available, we're going to go back and that plan will be revised and we'll continue to improve it. I really want to emphasize that this plan is, intended and will be transparent. It'll be an open process. Although this might be new to several folks on this meeting, it's definitely designed to leverage not only existing partnerships, but develop new partnerships and build on the current collaborations and knowledge base that we have in Rhode Island. We also will incorporate multiple approaches, and there will be multiple opportunities for individuals to participate. Those may include in-person meetings, they may include webinars, workshops, or surveys that's administered. And right now, because we're not quite done with our finalizing our planning process, and kind of keep those cards shielded, and you'll just have to, you'll just have to stay tuned if you want more information on that. I definitely, if anybody's interested, please, by all means, you can email me for more information. But also know that there'll be, this information will be widely shared as we get closer to starting that kickoff. And with that, I'm just going to flash is just on the work that I'm presenting today, and that's going to be part of the second presentation. There's a lot of partners and contributors and funding sources. This work takes a village. We're really fortunate to have such great partners and collaborators. And if you need any more information or have any questions, please feel free to shoot me an email. And with that, I'll kick it back to Skylar.
Thanks, Eric. So if anyone has questions regarding Eric's presentation, we're going to do Q&A at the end of this. But up next, we're going to hear about a project that has come out of our shellfish research group here at Roger Williams University. And so Ed Cabral, Rob Hudson, and Susanna Ozinski will be speaking. I also just want to put a shout out because I see Matt Griffin in the participant list, who was here at Roger Williams for about 10 years and helped sort of start this project way back when, but you'll hear more about that in just a moment from Ed. So Ed, when you're ready, we have your pictures on one of the first slides.
Good afternoon, Skylar and everyone. Yeah, so the project that's been referred to in here in the town of Warren, we started talking about it in 2015. The chairman of the Harbor Commission had seen an upweller system in Wellfleet where he was interested in seeing if the town of Warren could participate in a municipal project where we would grow oysters in an upweller and possibly put them out for open harvest in the area waters. So shortly thereafter, I made contact with Dale Levitt, professor at Roger Williams, and Matt Griffin was his assistant at the time. And we put together a plan, and actually they had an upweller system that they donated to us. And we got started in the spring of 2016 and had the upweller system running by, I believe, the first batch of oysters went in that upweller later on that year in September, I believe, and we actually grew them quite quickly, and they were put out in an area in the Warren River in November of 2016. And we've done that every year since. And just with an effort, it's kind of a dual effort. We're trying to do some education here with the local students in the school system in the Bristol Warren Regional Schools and the public. We've posted a pretty informative sign at the location showing what it's all about. And it gets a lot of interest from the public that visits the town wharf. It's at our municipal town wharf on Water Street. And Roger Williams, like I said, they've supported this project every year. And they're now with the new staff with Skylar and Rob and Susanna, they're doing some extensive studying of this project and the oysters that we've put out and we've been putting out. So, I'll leave the science part of that up to them to explain, but so far it's all been good here on the town end. We're curious to see how productive it ends up being. And we've been 5 years running now and appreciate the support of Roger Williams. And I know the general public in the area supports it. Warren is known for oysters and its history from the early 1900s. So people around here would like to see it, return to a big booming area with oysters. So obviously there are benefits that the oysters bring to the waters that also people are interested in seeing, that if they could clean up some of the waters and have those type of side effects, but that's the town's reason for being part of this. It's a municipal project that we donate our time and energy into monitoring the oysters, raising them during the duration that they're in the tanks each season, and then putting them out in the local waters. Eric was instrumental in getting the area that we put them in off Jacobs Point to a limited take area. Unlike other projects, I believe that this one, we put them out in open waters. Takes quite a while for them to grow to legal size. And then once that, that area stays open, they're open for harvest by anybody. So there's a reduced limit, there's one peck restriction on the take. So try to give them a chance to grow and hopefully spawn some new seed. But again, that's the study that Roger Williams is handling for us and seeing how that goes. So, and that's really the town's end of it. We're happy to be part of it, and hopefully it produces something good for the future.
All right, thanks, Ed. So let's see if I get the right screen going. So hi everyone, I'm Rob Hudson. I'm at Roger Williams University. And Roger Williams University has done a whole host of projects over the years. And the ones that I'm going to be focusing on today is just two of them, the ones that Ed was just referring to. So the first one is the Hasson Fellow Community Project. Again, this is started with Dale and Matt. And it was basically the RW Shellfish Group, and we partnered up with Ed and the Harbor Commission, and we worked with them. And the goal of this one was all here is they were rebuilding the bulkhead, so we had to reestablish their upwaller. The photo of the upwaller is right here on the right-hand side. And so we need to get this upwaller back up and running. We need to give them the seed that Ed was talking about. So that's the small oysters. We said that we would give them around 2 millimeters in valve length at that time. And they would maintain it, with some of our assistance and guidance, maintain them at that upwaller until it was time to bring them out to whatever location. Those locations were up for grabs initially. There was a couple of locations that we were looking at in the Kickoff and in the Warren Rivers. So we would kind of let them know where would be the best place for some of these oysters. Ed's mentioned it, Eric's also mentioned it. What are some of the benefits of doing this work? So we have enhancement of the actual shellfish populations directly within the study sites. Water quality, Eric's talked about a lot of the ecosystem services provided, water quality being one of them. And then again, we were looking to provide a recreational shellfish source of oysters, in this case, to the people that want to get out there and actually just go out and grab a quick bite to eat or some local shellfish. How do we determine if this is useful or working? So our monitoring metrics, both growth and survival, we looked at 2 different stages, or we're going to be looking at it at 2 different stages as well. And that's within the nursery system. So there's 3 phases when it comes to shellfish aquaculture. There's the hatchery, there's the nursery, and the grow out. This would look at the nursery and the grow out section. So the survival is when we place the animals inside of the upwaller, how well do they survive from the upwaller when we first place them in there to the day of removal. And then also after we're placing them out at the within the shellfish sanctuary. So that's the survival, the 2 different time points of survival. Same thing happens for growth. What's the growth inside the nursery system within the upwaller? And then what is the growth of those animals out at the sanctuary site? And all this data is being collected in order to gauge how efficient the utilizing the upwaller is, and how efficient is all this in actually enhancing the town waters with oyster seeds. A little bit more press on the right-hand side, similar to what Ed had just shown, those 2 articles. Here's a 3rd article that was released around the same time. And the image to your left-hand side is the signage that we have actually sitting right outside of the upwaller at the town dock. So even if you didn't make it to this presentation, if you happen to stumble across the upweller, you would be able to go there and you would get a good rough working idea of what is happening right on the side of you inside of that upweller. So on to the next project. This one is by the Shellfish Restoration Foundation of Narragansett Bay. And if I gave you enough time, you would read and you would see that this is the exact same slide as the Hasson grant. All the goals are basically identical to the Hasson project that we proposed in 2016. And if the goals are the same, so was the benefits, and how do we monitor it? So why did I just show you 4 sides that were the same? This is the difference. In the Hasson we proposed single set oysters. So if you look here at each one of these dark spots on this cutting board are single set oysters. And that was what has been placed inside of the upwellers since 2016. And on the right-hand side, these other 2 images, this is what spat on shell is. So Eric had actually already talked about spat on shell. This is a bag of culch, and inside of it is, like he was saying, it's either going to be surf clams or oyster shells, something that's been seasoned and conditioned. So state law requires us to season it for 1 year, basically slough off any old material that's on it to make sure that we're not transferring pathogens around from different locations. And then we had basically a truckload of oysters spat on shell bags. And this is what you get on the inside. So this red arrow right here is pointing to a single oyster on top of a surf clam shell. So you can see that there's a larger cluster of oysters on this shell. So each shell has multiple oysters on top of them. Well, yeah, each shell has multiple. All right, so why are we switching from, or why are we comparing single set versus the spat on shell? Is the spat on shell provides additional ecosystem services that Eric had talked about. And I'll highlight exactly which ones they're a little bit different from the single sets to the spat on shell in a second. The other thing is that we're hoping that they'll be sticking around for harvest. As Eric and Ed has mentioned, it may take a little while, but they'll get up to size and they'll be able to be harvested and consumed by the residents of Rhode Island. So that happens in a couple different ways. One is there's all this extra shell material from the culch, the old shell of surf clams or whatever. We're hoping that that gets locked into the sediment, and then that will hold it steady as the currents are going through, as any storms are moving through the system. We don't want it to just throw it on the shore or take it into deeper waters, or just completely disperse into locations that we can't find. So that's one of the hopes, is that the spat on shell would be locked into that position versus the single set, which is not able to fully lock into it because the animal has to stay out in order to filter feed. And all that shell also provides a lot of habitat for other animals, as Eric mentioned. So it's harder for a crab or another predator to get to the food, the oysters, with all those other animals around it. The highlights in red are specific more to the spat on shell animals. All these ecosystem services on the screen right now could be found whether it's a single set animal or a spat on shell animal. But the idea of that animal locking into the substrate helps again with storm surge protection, stabilizing the submerged lands, and shoreline erosion. I mentioned that it's hard for the crabs to get to a single oyster once there's a culture of them and there's a bunch of them grouped close together. The other thing is it's providing a nursery habitat for many of those other finfish species that are both commercially and recreationally important for us here in Rhode Island and wherever these types of systems are being deployed. So zoomed out view, that red boxes are where we're going to zoom into. We're here in the Warren River. The green polygons at the top, that is where the spat on shell sanctuary was for this past year. The red box is where the location where the upweller that I had already shown in a couple of slides previously, that's where that is located. And then what was happening on the delivery day of the spat on shell is we pull up with a truck, we take out the bags of culch, we cut them open, we place them inside these envelopes. These envelopes are then placed inside the cages. And then the cages were deployed in those green polygons. Maintenance. This is where the fun comes in, more in this case, more for Ed and his team. Lots of work here. So each one of these round circles are silos, and inside those silos, if you look in this photo right here, is a bunch of baby oysters sitting on the bottom of a screen. So what an upweller is it's a nursery system that force feeds the animals nonstop, 24/7. So the water moves up by the animal due to using pumps and equilibrium that we're able to basically force feed the oysters the entire time that they're inside of there. There's a lot of maintenance that typically goes for with an upweller system. Like every single day, somebody should be stirring up all the shellfish inside of there. Every other day, a partial cleaning. Every week, a complete tear down and cleaning and maintenance of the system. So a lot of maintenance goes into this. The cages actually ended up in the spat on shell stuff actually had a far less maintenance than what we originally anticipated. We were supposed to go out there once every 2 weeks, and then that increased to like once every 3 weeks with the same results. And we placed the cages out there, and you can see that this is what they look like during their initial deployment, and this is what they look like at the end of the season. So there was not a lot of fouling from either algae or tunicates or the colonial solitary. So a lot of the fouling organisms that would prevent water flow going through there and prevent the growth of the animals is what we're always concerned with. We want to make sure that there's plenty of food going, water moving by those animals. That way that's plenty of food flux. That's a lot of food that they can take in and a lot of their waste being removed. And that makes for a happy, healthy animal that grows quicker. We have here the triangle on the right, the red triangle on the right is of the shellfish management area. And we have the white polygon with inside of it is the study area for the shellfish restoration funds. And what we did was we took the single set animals, and we have both the north and the south site for single set and spat on shell. And we're trying to divide it up to kind of get a rough idea of how well they survive throughout this coming winter. So we have already did a baseline survey. We've checked out the entire area, and then we deposited. You saw images from some of Eric's photos of basically deploying spat on shell material. And we did the same thing with the single set, which was like that for those newspaper articles showing Matt and others throwing the seed out. It's similar to that, except for we, instead of throwing it out there, we kind of put it around these lat longs in order to monitor it after.
Hi, everyone. My name is Susanna. I'm going to give a brief little rundown about how the grow out timeline looked. So we have our timeline with the spat on shell that you'll see in the green and the single set that you will see on the white. So we initially had the oysters spawn, and we wanted to get about 100,000 individual oysters to spawn onto all of those spat on shell poach bags. And at the size we were looking for was 250 microns. Those were grown for about 2 more weeks, and then we moved them into those cages you saw on the previous slides. They were put at the Warren bulkhead, and those bags were put into individual cages. The cages were then just left to kind of grow out. And like Rob mentioned, they were checked on every once in a while to make sure, everything was going all right, that the biofouling wasn't getting out of control or anything like that. And then a week later, on July 3rd, we got our single set seed, and we had approximately 100,000 oysters that were at about 5 millimeters in size. And we put them into the upweller that you also saw pictures of at the Warren dock. They would grow for about the next 4 months, and they would be taken weekly by volunteers from Warren, as well as being checked on by some of the people from Roger Williams. They would be rinsed, they'd be washed and scrubbed down just to make sure that fouling wasn't building up in the upweller and on the silos as well. And then on the 24th of September, the spat on shell cages were emptied out. The shells were deployed at Jacob's Point, and they were just put right along the bottom where they would grow for the next couple of years until they're able to be harvested. And then about a month later in October, we did similarly with the single set oysters. We took them out of the upwellers. We would do a couple different measurements so that we could look at that growth and that mortality that Robbie mentioned earlier. We ended up with a final count being around 75,000, which was a little lower than we would have liked, but we had a lot of growth. As you can see, the sizes now were over 12 to 22 millimeters, which was great to see. A lot of growth was coming out of those upwellers. And they were similarly just put single right out, no cages, no bags, and they were placed on the bottom alongside where the spat-on shell was located, as you could see in that previous map picture. And they will be left there for the next couple of years.
Thank you. So what we have here is a quick sneak peek. Of course, this project is still ongoing, and we have the on the left-hand side, these are the single sets. This is about the mid-class that Susanna was just talking about. This is an image for that. We're going to use a software program called ImageJ to really hone in on the average sizes and growth of the spat-on shell. But you can see right here, this is about 20-25 millimeters in size. And when we first deployed them, they were maybe 300 to 500 microns in size. So they definitely grew. So basically they went from about 250 to 300 microns to 2,500 microns and so forth. So that, just to give you a rough idea of what the size difference is from start to finish. As Susanna said, there's always going to be some type of mortality, but overall it was still pretty good. So in, I don't know, 10 plus years that I've been doing shellfish restoration since 2005, so more than that. Anyway, so a lot of people ask you what you can do to help, and the main answer is just being a good steward. So, you know, everybody wants to protect. We were talking about having a resource for people to go out there, and as a Rhode Islander, we all want to go out there and grab our shellfish, enjoy it at some point. So the picture on the right-hand side coincides with the top link, and that is what I just took off the DM website today. Is, you know, we're in a management area. Make sure you know how much you can take, you know, as you're taking out your family members and training the next generation of Rhode Islanders how to enjoy that. This is, you know, being a good steward that way. The other thing that's not on this list, which I did know that there was a season, it took me a little bit longer to find it. There is a season for oyster harvesting. There is a season for bay scallop harvesting. So just make sure you know the seasons and teach the next generation the proper way to enjoy our resources. And, you know, you got to thank the people that made this possible. So definitely the volunteers, the RW students, and other staff members that are not here tonight. Matt's here. Thank Matt and Dale and Brad and so forth. We also have the Warren Harbormaster. You just met Ed. His commission members that are out there and taking care of those upwells is a huge help for us. Funders of the Hasson Field Community Project and the Shellfish Restoration Foundation of Narragansett Bay, and we're always in restoration projects. We're a partner with DM. So thank you, everybody.
All right, thank you, Ed, Robbie, and Susanna. Just one more plug for the Roger Williams program. If you happen to be on Instagram, we started a relatively new account if you want to follow some of the pictures that we put up with what we're doing. So now, from what I understand from Azure, we are in the Q&A section. Azure, do you want me to just read off from the chat and then the presenters, if they feel that they can answer the question, will answer the question?
Sounds great, Skylar. Thank you.
Okay. So the first question I have in the chat is from Don Libby that says, what is considered for suitability index? I'm assuming that's habitat suitability index. Physical site / underwater survey, historical shellfish data, local stakeholder input. I don't know if that, Eric, you might want to answer that first, or.
Yeah, thanks, Skylar, and thanks for the question, Don. So different projects have used different approaches, but you're pretty spot on, at least in the initial phase. And what we're planning to do is a number of physical and biological attributes. And so, like I mentioned briefly during the talk, We're conducting survey work presently, and we will do that at least over the next year as part of our partnership with the Nature Conservancy in Northeastern. And some of the attributes or factors that will likely be, will definitely be considered in part of the Habitat Suitability Index. And mind you, this is just a suitability index for oysters. There's a whole nother level when you can bring in other factors, more for the larger, let's say, restoration suitability analysis. But in terms of just the oyster suitability index, aspects like salinity, and that can be temporal measurements, that can be the average salinity for the year. You can focus on the salinity during critical times of the year, such as during the spawning season. Substrate, water depth, temperature, again, there can be temporal aspects, whether there's a max or a min, the average, dissolved oxygen, turbidity, certainly oyster presence or historic oyster presence, whether there is evidence of oyster scarring, shell, or actual living oysters present. Other studies that have had abundance of predators, disease, fouling organisms, all of those aspects can be considered, but of course, with each level, the amount of data requirements increase.
Okay, thanks, Eric. The next question, and Bob put in some conversion factors for us. Thank you, Bob. Next question is from George Davis. Are you able to say how the Block Island Suitability Index compares to the other Rhode Island locations? It's a specific question.
Sure. So right now, at least of the information that I'm available, the actual oyster habitat suitability indexes are largely restricted to the coastal ponds. That doesn't mean that we can't create a limited index based on subaqueous soils or other data, but and George, if you have one from Block Island, by all means, I'd love to see it. I haven't seen one that's been developed for Block Island, but I do know that Nrcs, Rhode Island Nrcs has conducted subaqueous soil surveys of Great Salt Pond. And so typically in a scenario like that, that would be the first information that we would look to is soil data. If there's salinity, and sometimes you can infer the salinity, there's locations where there's marshes, freshwater inputs. You can look to marsh or terrestrial vegetation sometimes, or walk around with a salinity meter just to get a feel for whether or not there are some general variations in salinity. But to your question specifically, George, I'm not aware of a specific HSI model for Block Island, but if you have one, by all means, share it with me. We'll consider it. And I'd love it if there's one that exists.
Okay. Next question. What is the average attrition rate of oysters from spat to harvest? Which I think maybe all three of you might have something to say about that, but.
I'm happy to address it, but I'm also happy, Robbie or anyone, if you guys have collected data from your surveys, or if you want to just talk about it first, by all means, but otherwise I'm happy to talk to it.
You want to take the first crack?
Sure. So what we've seen from some of our restoration work, again, it varies by system. In some of your more saline systems, there can be other factors that contribute to the growth rate of oysters. Certainly the lineage and how well matched that lineage is to the environment that it's in, and whether those are hatchery lineages that are more, that have been reared over time for certain attributes that are good in an aquaculture setting versus a natural or wild lineage. There's lots of factors that can affect growth rates. We've seen growth rates that would suggest in some cases it can take 3 years And that's starting at, these are spat and shell oysters that are typically deployed, at least in the scenarios that I'm talking about, somewhere is between, 1.5 to, let's say, 2.5 centimeters. And thus over 3 growing seasons, they can reach maturity. There's certainly reefs that we have that have been able to do it in 2. And there's, but in generally it's 2 to 3, some lag a little more, some are a little faster.
Yeah, so like Eric was saying, that there's definitely different factors that affect the rate of growth. We have the lineages, like you said, we have Rhode Island lineages versus something that is selected for a hatchery. A hatchery will handpick specific oysters to breed further in a hatchery that they normally connected with vertically with inside their own company or business, typically. Unless you want to take something off your farm and then send it over to a hatchery specific for a private spawning event. But anyway, so the hatcheries are growing these things specific for aquaculture, are growing for, maybe some colorations or stripe patterns on the shells. They're going to be growing for how fast they are. Some of them could be growing for different being resistant for certain pathogens that are and diseases that are in the water that typically affect the bed. So there's different things, different trade-offs that people look for. And then what's going on in the water, like Eric was saying, water column and naturally some of those lineages, like some of our native lineages, like the green hills seem to be pretty small. The narrow rivers ain't that much bigger. They're naturally smaller oysters than something that you would find in like Connecticut or something.
Azure, we're almost at 5. Do you want to continue with the Q&A that's here for
I would
now?
say yes. Yeah, folks did put the questions in, and we recognize folks might have to hop off right at 5, and that's fine too. But let's get to those as they're posted. Thanks, Skylar.
So the next question's from Ayana Melvin. The Warren presentation mentioned involving youth in the process. What specifically did youth engage in, and others encourage you to participate in this kind of work? And Ed, that might be a question for you, I think.
Yep.
Yeah.
So one of our Harbor Commission members, Roxanne Wall, has been very instrumental in helping out clean the tank every week and all of that. He had connected with the science teachers, I believe at the 4th grade level, starting there, trying to get them to get kids participating and or at least coming down to see the upweller and explain what was going on there. Unfortunately, the season for the growing comes to the end of one of the school seasons and then the beginning of the next school season. So hands-on wise, we haven't been able to coordinate anything hands-on. We've tried to get them involved in, maybe helping us deploy the oysters at least. Obviously with COVID, things changed in the school setting. So that kind of fell apart for the last couple of years. But I know the 4th grade science teachers were, I don't know how involved they ended up getting the kids. They never interacted with us during that. Again, because it was just kind of an offset season for the school. But that was one of the interests. Was to try to get the youth in the area, being, Warren surrounded on 3 sides by water here. So we try to get the youth to understand what's in the waters and get them involved in participating in, the future of protecting these waters and what's there. So that was a driving force. And like I said, Rock was the one who was working with the school department trying to get that accomplished. But unfortunately, like I said, hands-on wise, we weren't able to accommodate anything, specific.
Thanks, Ed. The next question is from Emily Skihan. And this is for Robbie and our group. She doesn't see a lot of information online about the Hasson Community Project or the Shellfish Restoration Foundation of Narragansett Bay. Does Roger Williams plan on scaling up this restoration project to potentially other areas? I think I can answer some of this because this is from our group, but I think that this project's been really successful thanks to these funders, which may not have a big footprint on the web, but were really instrumental in these projects. And I think we would like to in the future do more projects like this, but I think it depends on what communities want and who's interested and engaged. We have another upweller related project over in Warwick with the Rhode Island Shellfish Association. And that's been going on for a long time. Again, another project Dale Levitt helped start, and they seed those near Spawner Sanctuary. We just helped them in November seed them. So I think we would love to do more projects, and it's dependent on funding, both for our time and materials, and finding willing participants and making sure that those projects fit the needs of the community. I don't know if Rob or Susanna has anything else to add, but that's sort of how I see potential future projects.
That sums it up.
Yeah. Next question from Dwight McNeil. How can your very good work be scaled up to have a significant impact on the ecosystem, as with the Billion Oyster project in New York? I don't, Eric, that might be a question for you, I think. But
Sure, yeah. Great question, Dwight. And I assume, maybe not, but those who didn't see it, there was a really good article in the New York Times Monday. I think it was Monday. Hard to keep track of time. It could have even been last week. But either way, getting back to your question. So that is one of the, one of the primary purposes of the Shellfish Restoration Plan. It doesn't go in with a default, let's scale this up as New York is doing, and it's really going to rely on stakeholders within Rhode Island to inform the process and tell us what are the restoration priorities, what are the services you want to see and spatially where, and that type of information can be used in a larger analysis to say, okay, in this area, these are the types of services, the type of work, the level of support that we have for it. We can use the habitat suitability index along with the other spatial coverages regarding marine habitat, other human uses, to try to whittle down where that work can occur. And then we can try to estimate how much work do we need to undertake to achieve that goal. And so I'll flip it around and say, I think the shellfish restoration process by design offers us a path forwards to certainly expand restoration work throughout Rhode Island. However, there's obviously going to be spatial considerations based on what the restoration priorities wind up being, as well as the ultimate suitability for doing that restoration work, which accounts for a number of factors.
Thank you, Eric. I don't know if Robbie or Susanna has anything to add. But I was going to move on to the next question from Lori Cronley. The Narrow River seems to be an oyster success story, though it's closed to shellfishing. To what do you attribute Narrow River's success?
Eric should probably take this one, but overall, it is close to shellfishing, so there's no harvesting pressures on it. We believe there is some type of resistance to some of the common pathogens, such as dermo, MSX, and SSO, which are all pathogens that negatively impact shellfish populations here locally in Rhode Island. So we use them in some of our other research projects as a marker for Rhode Island lineages that we can put forward and test out, whether it's for restoration sites or for farms. So there is some nice components to that. They also, the flushing and there's other, like the environments, the habitat's just right. Eric was talking about the habitat suitability index. If you chalked it up, a lot of this area would be pretty prime habitat and have received some high scores. The flushing is just right. It's a fjord. It's not a typical flushing of Narragansett Bay. So the water that going in and out of the Narrow River or some of the other pockets of wild populations that we have in Rhode Island is completely different. So there's a lot of components. It's like a perfect storm of good things that happen for the oysters that are still within the Narrow River.
Yeah, Robbie, you you did an excellent job explaining that. So thank you. I'll just add just a couple of things, and most of it's redundant. You know, like Robbie said, most of these systems, and in particular the narrow river, it's a combination of factors. So you have the lower salinities throughout most of the upper stretches of the narrow river where you find them. That it's been closed to harvest for a long period of time has allowed that population to assist and probably adapt locally to those conditions, at least that it's experienced over the last, several decades. Robbie spoke about the larval retention, just the hydrodynamics of that system we assume a lot of the larvae to be retained in these upper ponds. And so it is a really, there's suitable habitat around the edges, the temperature, a lot of the things that we talked about as attributes that can be considered in the habitat suitability index and that Robbie named. I think collectively they interact to try to allow for some of those populations, or at least those populations, to persist at a higher biomass. I would say the folks that are interested in the narrow river system, you should also consider reaching out to our local Rhode Island Nature Conservancy chapter. They've been doing work in the narrow river, for a number of years. And so I'm sure they have some additional insight and would welcome the opportunity to talk to you about that. And if you don't have you can go to their website or shoot me an email and I'm happy to connect you with them.
All right, so I have one last question and then we're wrapping up Q&A, but what role, if any, this is from Nick Carr, what role, if any, do you see privately operated oyster farms in restoration at large?
I have something I might add to that. So in the Chesapeake Bay, if you might have heard, they do have certain credits similar to how you would see with like big farms with nitrogen credits, where the farmers that grow to sell to restaurants and bars and such are also able to help in restoration with the environment. That's something that people are interested up here in the northern side. It's a little bit more of a work in progress than down south, but with farmers, they often are working to sell their oysters, right? So that's usually their main goal. So they're usually located in areas that are either always open or the waters are clean enough where they can sell the oysters without problems. Or if they are located in conditionally or seasonal waters, then they have to work with the laws a little bit more. With restoration, you often see them located in areas that, yes, are seasonal or open and approved, but it's usually areas that might need a little extra help where the waters aren't clean and can't necessarily be used to consume. And that can cause a little bit of a problem with farmers. If their main goal is to sell oysters, then it adds a little more difficulty into also trying to grow oysters to help out the restoration site. I think that's a big thing with farms, but I'm sure there's other roles as well that they can play to help.
Any additional comments?
I'll just tag on one additional thought, at least from the division's perspective, is that, and Nick, if I'm not addressing your question directly, definitely feel free to follow up with me. But for all of the recent work, at least for the two projects that I've shown during the presentation that I gave earlier, aquaculturists were a key, component of that. They, without their knowledge and their ability to grow shellfish up to a size that, is able to be deployed in these restoration settings, without their really spatial distribution throughout the coastal ponds in Narragansett Bay where we're conducting shellfish, we would not be able to do this restoration. And it's really their knowledge, their expertise. We've gained a lot. They've provided insight not only to locations, but lineages, different approaches we should consider, and we've adapted, to the degree that we can, some of our restoration practices, in response to those suggestions. So specifically, they're to date, they've been a key component And as far as I can see, at least in Rhode Island, they will continue to be, a key partner in oyster restoration as we go forwards.
All right, well, thanks. Azure, it's on to you now.
Great, thank you so much to everyone. And it looks like your fantastic work and presentation has inspired a volunteer. So William Ham is offering his services in any of this good work, and Warren beyond. So I just want to thank all of our presenters. Eric, your great work continues to inspire, and certainly Skylar for facilitating tonight and for leading an amazing team. Susanna and Robbie for your excellent work, and Ed for a collaboration that is really known statewide. And the town of Warren is the model stakeholder, town stakeholder to work with, and that's very appreciated across state agencies and university folks alike. So thank you all so very much. Please stay tuned. If you haven't already, join CRMC's aquaculture listserv that we are finding is a very great place to post webinars that are upcoming. We'll have one likely late January, and then on through February and March. So there's 3 more in this series yet to be planned, but stay tuned. And I just want to thank everyone for your great time. And in the next couple of days, we'll have this posted up on the BaseAmp website. So thank you all so much. Have a great evening.